everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 19 of Surfing the NASH Tsunami. Our look at the retrospective analyses in the NAIL-NIT consortium and some other news starts now.
This week on Surfing the NASH Tsunami.
At a high level, NAIL-NIT is really about two different projects. Retrospectively bringing data together, breaking down stovepipes, and linking investigators, sponsors, patients, and regulators and investigators all together to mine the data that's already been generated. And then the second part of this is taking what we've learned from that and then building the ideal prospective trial to evaluate these non-invasive tests for the intent of prognostic implications.
Can we combine data from 5, 6, 10 clinical trials and look at them and see how they perform in comparison to a liver biopsy, but also in comparison to each other and some of the scores that we've developed? Can we also look at the placebo response rate in different trials and try to get a deeper understanding at what's causing this variability?
The whole point is most people that get to a trial are the very few of the very few of the very few, and we want the whole of the pie to be diagnosed.
That's for the future, and really to get there, we cannot just keep jabbing and stabbing people on the right side of their body with a liver biopsy.
If we accelerate this and move beyond the biopsy and have NAIL-NIT produce successful outcomes, we'll have many more study centers, even less experienced hepatologists being able to enroll their patients because they're everywhere, these patients, and I think we're going to get more diversity.
So there's your timeline. We need to have a very clear, easy to use, non-invasive testing strategy for at least a context of use of diagnosing the patient that would qualify for treatment in a very simple, easy to use, non-invasive format by Q3 of 2023, if not sooner.
Every week, a global community of fatty liver disease stakeholders comes together to explore the most important challenges in diagnosing, treating, and developing medications for patients with fatty liver diseases. Join hepatology researchers and key opinion leaders, Dr. Stephen Harrison and Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and this week's guests, hepatology researchers and key opinion leaders Drs. Naheem Alkhouri and Mazen Nuruddin, as they discuss their goals for retrospective analyses from the NAIL-NIT consortium this week on Surfing the NASH Tsunami.
Для всіх, хто цікавиться мережею NASH або загалом жировою хворобою печінки, ловіть хвилю.
First of all, let me thank Anatol Kar for reading us in Ukrainian again. This is a very special week here at Surfing the NASH Tsunami for a variety of reasons. As those of you who read your LinkedIn, Twitter, Facebook, or your emails from us know by now, this week is the second anniversary of this podcast. We recorded 2 years ago this week and dropped the episode 2 years ago on what would be Friday. And we thought we were going to do 4 episodes and maybe a couple of people would listen and Stephen would have a tape he could take around to some drug companies. And 2 years later, here we are. These numbers are pretty staggering. 126 episodes. 335 postings, 58,000 downloads, 105 countries. We mentioned those in the announcement. There are a couple of others. In addition to Stephen, Louise, and I, who served as the primary co-host, we've had 54 other panelists. We've covered 11 major events. And my trivia question for the day, for those of you who are listening live or otherwise, if you include Stephen, Louise, and me, before today, how many people have had their names announced as part of the podcast before we get to today's guest statement, since we started the podcast? And when you hear this on the podcast, but not live today, you will hear the Beatles singing Happy Birthday in the background because that tells you how old I am. So in the honor of second anniversary, we have a lot of really exciting news. First of all, we'd like to announce the formation of SurfingNASH.com, a new company, a partnership of HepDynamics LLC, which is my company, and HGTM LLC, company owned by Stephen. And we are creating SurfingNASH.com as a separate company, taking control of this podcast and dedicated to creating audio and visual programming and social media to keep putting the biggest dent possible on fatty liver disease. We figure we've taken a couple of kicks to the can so far, but nothing compared to what we're going to do now that we can really get started. So that's number one, and I can't tell you how excited I am that we're doing this. Stephen can talk about it in a minute. I'm just going to keep going because if I don't, I'll probably start choking up. I'm I'm way too emotional for my own good sometimes. Second, as we discussed, Surfing Nash is looking not only to continue the tsunami, but to start a whole set of audio and video programs to educate different stakeholders about NASH and NAFLD, looking from their points of view, their countries, their specialties, their issues, their everything, and helping to activate them to make diagnosis, treatment, and monitoring a larger part of their daily lives, including patients. Over time, we think this will include not only hepatologists, but every specialty that touches metabolic disease, allied health professionals, patients, maybe even payers. Last week, I announced that we've signed an agreement to develop a 4-episode pilot series targeted at endocrinologists and others for whom treating patients with type 2 diabetes is a big part of their practice today. Next week, we're going to announce the series name and the date for the first episode, but I will tell you today, the first episode will launch sometime during the first half in May, probably on a Tuesday evening at 10 o'clock in the eastern U.S. Still making sure whether it's Tuesday or Wednesday. If you are listening and you can think of a cool name with a surfing theme for a podcast about diabetes and NASH, send it on in. If we use it, I'll invite you on the podcast to talk about how you came up with it. And even if we don't use it, I intend to read all the names next week. Okay, moving on. In a Broadway show or TV series, the way they work is they do a pilot and then lead characters sign on for one season at a time. And fundamentally, that's what happened here, right? We did a 4-episode pilot. It did really well. We signed on for season 1. Stephen, I think, had planned to leave originally at the end of season 1, stayed on for season 2, and now we're to season 3. So, Stephen, would you like to take a minute and talk about your plans for season 3 and what podcast has been to date for you?
Sure. First of all, let me say I'm very proud of surfing the NASH tsunami and where we've come. I mean, just thinking back to when this started in the middle of the pandemic, I remember sitting down with you and thinking about how we could continue to fill the void of news and information surrounding fatty liver without having EASL and AASLD and many of the other liver meetings. I think NASH was the only one that continued in an in-person format throughout, in some form or fashion, throughout the whole pandemic. And it really hit me that there was this huge void in communicating data that we had gotten used to. And so we started the podcast, and very quickly Louise came on, part of that as well, Peter Traber at first, and then we kind of settled into a battle rhythm. We settled into a routine. And I remember we were told, was it somebody mentioned early on that after about 10 podcasts we would run out of things to talk about and it would just kind of go away.
It was Peter. He wasn't worried about how long he was going to stay on the podcast because it was going to run out before he ran out of commitment time.
That's right. And then it just kind of began to build steam, much like a locomotive, just kind of starting out with a chug, and then it began to pick up steam, and we rolled through year 1, and then through year 2. You mentioned all the countries that we're touching, all the downloads that have occurred. I love your comment a while back. You said, believe it or not, we're the #1 podcast in Macedonia or something. I mean, there was those little highlights that tended to just keep our energy going. And I think there's a big special thanks to you, to Louise, to all of the people that have joined the podcast and been huge in trying to elevate the podcast. 3 of them are on this podcast today. Mazen, Naeem, Jörn. I think they've done more episodes than any other podcast member outside of the 3 of us. And Donna Cryer. Yeah. So again, I special thanks to you guys for taking your time, your effort, your energy. You're as busy as I am, and you'd come on the podcast routinely. And I just want to say thank you for that, and for the producers, for all the people that make the podcast go. I mean, it's amazing to take what we talk about today and then listen to it and see how it's transformed into this very professional, well-done diatribe of different things that we talk about. For me, moving forward, many of you know that I'm involved in lots of things centered around trying to find our first cure for NASH and driving the disease awareness campaign around NASH. And I want to continue to do that. But I'm going to take a step back and focus some energies in some other places. I'll still be a part of the podcast, maybe just not quite as often. I still want to try to make at least 50%, if not more, of each episode. So I'll still be around quite a bit. I'm so thankful to have Jörn stepping in the lurch here and filling that spot. He has been a huge supporter of the podcast. He brings lots of talent, lots of energy, enthusiasm, and passion, and a unique perspective that I think we all enjoy listening to, not to mention the German accent that helps. That's nice. That offsets Louise's English accent some. So anyway, I'm thrilled to still be a part of the podcast. I'm thrilled where it's come so far, and can't wait to see where it's going in year 3 and beyond, as we really are a conduit for content delivery. You mentioned all the other bits and pieces of information that is going to come out as a spinoff of the podcast. I think those are terrific additional conversations that are going to be had. But maybe I'll just stop there and say thank you to everybody.
Stephen, first of all, we all want to say thank you as well. And I'd like to invite everybody to say thank you at 2 levels. Number 1 is just say thank you and to talk about what this podcast and your role has meant to them so far. But second, Mike Wilson, our audio engineer, came up with this really cool idea called the Stephen Harrison drinking game, which is there are things that you just say a lot. Now, because this is NASH, the drinking game has to be done, as Naeem would remind us, with black coffee, with non-dairy milk if you need a sweetener. Yeah, that would be one of them. Hashtag real talk. That's right. So it's a drinking game with black coffee, a non-dairy sweetener, a non-dairy milk if you need that, and a friendly glycemic index sweetener like agave. So every time Stephen says any one of the things that we're about to note, if you're listening or if you're on, you're obligated to have a drink of coffee. With that, brave one, go first, just to say thanks and talk about what this podcast has meant to you and Stephen's role has meant, and then share with us a drinking game item.
I'm going to jump in first. I just love all of these ones, and I want to get it in first. Keep it simple, stupid. I just love it. I think it is the mantra of really explaining things to patients and other physicians and other nurses, so it comes across that entire set. So for me, keep it simple, stupid is one that I will always take with me. And I think my other thing is, despite not ever meeting you, I consider you a friend. And you're not getting out the deep fried turkey.
Oh no, no doubt. Yeah, keep it simple, stupid. KISS principle. That takes me all the way back to when I was a second lieutenant in the Army. I was told very early on to, don't make it complicated. Keep it simple.
And healthcare has become way too complicated, so we need to rein it back a little bit and keep it simple.
That's right.
I guess I'll go next. Well, first, happy anniversary and congratulations on all what you've accomplished. This is really amazing. This podcast has been really important for us in the field. I mean, there are deep discussions that go into non-invasive tests and endpoints for clinical trials and regulatory issues and pathology issues, and you're making an impact for all of us, and it's a platform for us to have these honest discussions about things that we think about, we talk about, and sometimes, we don't even have the data yet to support it, but, it generates a lot of thinking, and sometimes it's the way we start research projects. I've been listening to this for the last year and a half or so religiously, and congratulations. For Stephen, I guess for me, it's is the juice worth the squeeze? This is the classic Stephen saying, and I think, it's really important in the NAFLD field because it's a multifactorial disease. There's so much going on, and you need to always think about this. Whatever you're trying to do, is it worth it? Is it going to lead to moving the field forward? And this is one thing I learned from Stephen. So always, think, whatever you're doing, is it worth it? Is it going to change the field for the better?
Excellent. Miles, you want to go next?
Sure, sir. And just want to comment on the drink first. Rather than coffee, can we make it Irish coffee since I'm in a vacation mode?
You can do that, and your liver will attest to itself. It's fine.
Well, the first time I don't remember how I came across the show, but I do remember the first episode I listened to it. I think it was during a conference or something that I missed, and I just downloaded the thing and I listened. I'm like, this is awesome. If you miss anything, you will get this. It will tell you what happened. It will analyze it, and you will get experts in the field talking about it. I think that was an episode where Peter Traber and someone from the FDA were on. I think that was probably my first episode listening to it. And since that, I came on. I enjoyed the discussions, as Naeem mentioned. I enjoyed the challenge. I enjoyed moving the field forward. And I also enjoyed the social aspect of it, that you, Roger, do a great job. I mean, I got to know a lot of my colleagues, their children, what do they do, what do they eat, what do they drink. So the NASH world become like just one small world. Let me shift to Stephen. Where do I start? Well, the first thing crossed to mind, and I think he has said it on the show, and he and I are friends now. He gives you this certain military sentence that is as following: well, we're gonna get it done. And sure, things have gotten done in the last 2 years, in the show and outside the show, in the field, because Stephen and his vision and action, so we're gonna get it done.
Get her done. No doubt.
Which what we're going to talk about also today, the NAIL-NIT that is going to get it done.
Amen.
Absolutely.
So, Mazen, first of all, to be honest, thanks so much for doing this from vacation. The fact that you wanted to be on this episode and you happen to be on vacation doesn't in any way lessen the fact that you're doing it from Spain.
And I missed a Michelin-listed restaurant with my family. So that's how much I love you, Roger and Stephen.
Wow.
And you know what? We'd be a lot more impressed if it wasn't 4:45 in the morning where Louise is right now, but it's very impressive.
That's true. I have to give her that.
Very impressive.
Thank you.
Okay.
But you would never know that it's 4:45. She looks just as chipper and as dapper as ever.
Yeah, she does. She looks fantastic. Jörn, I'm going to let you go last. I'm going to go next, in part because I really like the one I've got for Stephen, and in part because you should have the floor for a couple of minutes, my friend. Stephen, first of all, I've said this before, but I'll say it again, an exceptional debt of personal gratitude. I was kind of wandering around at a point in my life trying to figure out what was I going to do next that had meaning and had impact, and bumped into you at hashtag 2019, and you started talking about making a big fat dent in fatty liver disease, and it just resonated. Took us a year to figure out what to do, and then it wound up being a lot more and a little different than what we had in mind, but here we are. And I had forgotten how much fun work could be until this thing really took off. So I owe you an exceptional debt of gratitude for that. I've said that before on this podcast. I've said it privately. I'll say it again. Everybody that does this is infused with your spirit, although we're going to have to decide who takes on the role of commenting on the furniture in people's houses when we see them. I mean, Manal is moving to Minnesota. She's going to have a whole new house for someone to comment on. So you're going to have to show up when she's on so that you can talk about her furniture. I don't know anybody else who could do it justice.
Sure. Absolutely.
Here's my Harrisonism. I think it speaks to the ambition that is in front of us as well as where we are right now, which is we are flying a Wright brothers plane when what we really need is a fifth-generation strike fighter, right? And I think that gets Nash in a nutshell.
Nash and NITs and all of it, right?
Fatty liver in the broadest sense, I think that totally captures it. So that'll be mine. So the reason I wanted to let Jörn go last is that Jörn will have the thoroughly enjoyable, but in some ways not entirely enviable task of sliding into Stephen's chair. And I've been scribing him for a while as the fifth Beatle. We're dropping the fifth. And Jörn, starting this week, will be a permanent member of the podcast. Stephen will be with us. The target is half the time, maybe a little more if we can pull that off. Probably more of these other two gentlemen and some other folks who have been with us. But Jörn, my friend, for the first time as like the official key opinion leader of the podcast, the floor is yours. Talk to Stephen, talk to us. Have at it.
Well, thanks, Roger. And clearly, I'm not taking Stephen's chair. Wouldn't make me comfortable sitting in it. So I'll pull in my chair, and I'm happy to join you guys in this. It's been really a tremendous pleasure listening to the podcast. I said that before I started out running and kind of like Mazen stumbled across, I think it was the Complete Response letter where I thought, hey, I work on this. What are they talking about? And just got glued on and stuck with the podcast. And I'm happy every time I can come on. And now that you invited me to join on a weekly basis, even more exciting to see friends and discuss this. And I'm 100% certain that it will carry forward even if face-to-face meetings are coming back up, because it covers so much more content, depth, different perspectives, all the type of information you don't get during a regular short meeting. Looping this back to Stephen and you, and kudos for what you've created here. It's year 3 kicking off, really tremendous effort. There are many things I could say about Stephen. I tremendously enjoy the energy you bring into this podcast, Stephen. It's beneficial for the field and the things we talk about. So it's been a big joy and pleasure joining you here, and I'll be looking forward to you taking the seat because it's not taken up by me. Plan to bring in my own perspective, a little bit European, German thoughts, as a clinician scientist from this side of the world. Clearly, what's interesting me most is, of course, the end-stage liver disease patients, because they're real and they're in urgent need of some sort of therapy and NITs. And I guess my Harrisonism in this context is the shriek and peak. It's not cancer, but it's something very surprising, and nobody would have thought this person has cirrhosis. And here it is. Stephen Harrison would have probably told you before, but nobody looks at it. So I think it's a quote that says a lot about the disease, and that's why I like it.
You're bringing your chair, sitting in my chair, whatever you want to call it. I think you've always brought a nice perspective, a fresh perspective, one that is steeped in wisdom and knowledge. And whether I hear you on the Litmus Consortium or whether I hear you as a panel discussant at a scientific advisory board, or we're doing a satellite symposium together, or you're giving a lecture at AASLD, EASL, or NASH Tag, or any one of the other venues that you and I happen to be at together, I always learn something from you and like your perspective. Same thing goes for Naeem and Mazen as well. We're only as good as the collective we. Each of us brings something unique to the table, but together we're much more powerful and our voice resonates much louder when we speak collectively. And I think we all do a great job of that. So the best days of the podcast lie ahead.
I do believe that. And as we turn to NAIL-NIT, I just want to go back to Jörn's comments for 1 second, which is, Jörn, I've actually had the experience of watching Steven do a peek and shriek.
On which occasion was that?
Right before the pandemic, I brought some folks from Sonic Insights down to San Antonio to meet Steven, and I was the guinea pig. When whoever it was put the fiber scan on me, I've got very large rib cage. Steven, turning slightly green, looked at me and said, has anyone ever told you that you might need a liver transplant? And I said, no, I just have big ribs, try it again. And in fact, it turned out I just had big ribs, try it again. But for that one moment, I watched a little bit of green tinge in his face, and I said, I'd never heard the expression peek and shriek, but every time I hear it now, my mind goes to that one moment and your face.
So moving forward, the one phrase that I like to use that I think is apropos as we try to find cures for fatty liver, find non-invasive tests to define the 3 different contexts of use that we focus on, is we want to take a broken piece and make a masterpiece. And I think that's really what we're all about. All of us are driving toward that same paradigm. And the people that are listening today, they all want the same thing or they wouldn't be listening. So that's how I'll end the little segment on Harrisonisms, taking a broken piece and making a masterpiece.
So with that as a transition, why don't you lead into the discussion about through retrospective and NAIL-NIT analyses that Naeem and Jörn are driving?
Mazen might be the one to better jump into the details of NAIL-NIT. He has really been instrumental in dotting all the I's and crossing the T's as we began to put form and function around this concept of NAIL-NIT. But I do think it's worth maybe just a high-level thought. So I've been doing this for over 2 decades now, and there have been a lot of attempts made to try to bring together data and break down silos and stovepipes and begin to cross-pollinate, and through the power of numbers, reach some conclusions. Everybody has the will and the want to do that, but it's never really been done. There have been attempts at multiple different times over the past 20 years to do it with some degree of success, but really not to the level that we're hopeful that NAIL-NIT will achieve. And NAIL-NIT is just that. We want to nail the non-invasive test. We want to find the right test for the right situation at the right time to answer the right question, whether that's diagnosis, whether that's monitoring for therapeutic efficacy, or it's prognosis. We think we have accumulated a large body of data already that just hasn't been mined to its fullest extent. And then prospectively, we realize that we have work still to do. So at a high level, NAIL-NIT is really about 2 different projects. Retrospectively bringing data together, breaking down stovepipes, and linking investigators, sponsors, patients, and regulators and investigators all together to mine the data that's already been generated. And then the second part of this is taking what we've learned from that and then building the ideal prospective trial to evaluate these non-invasive tests for the intent of prognostic implication. It sounds like a big, fat, hairy, audacious goal, 2 of them, in fact, and they are. It's going to take a lot of work. It's going to take a lot of people willing to change the paradigm, to say what's happened in the past is maybe not work the way we wanted it to, and is there a better way? Is there a way that we can join forces and answer some of these questions? I think it can be done. We have, I think, a terrific team assembled to address this. We have, at a high level, we have incredible interest from sponsors, from pharmaceutical companies, from device manufacturers, from companies that are invested in diagnostic strategies, and I believe also, from the regulators. So maybe, Mazen, do you want to jump in?
Yeah, I'm happy to. Like, you summarized it very well, Stephen. And I just want to go back to the bedside. And this is where the whole thing started. I was frustrated when I had patients that their non-invasive testing, transient elastography, MRE, showed stiffness. They had elevated liver enzymes. I did my best to kind of narrow it down for them and then find them a treatment. And then I biopsy them to go for a clinical trial. And I'm like, oops, I'm sorry, you don't have ballooning. And the patient's like, ballooning? And then there were other occasions when the NAS score was 3, not 4, and they didn't get included. And I stumbled. It's like, what do I do now? Like, repeat the biopsy right away or wait a little bit and send them to another trial and see if they're going to read it differently? So one day I think we're walking in a hotel, and I think it was just after the patient, and I started talking to Stephen about starting the NAIL-NIT, and I was saying this sentence and he was saying the second sentence in my mind, and I think saying the third sentence in his mind. So I think it's the wish of all people in the field. What I want to acknowledge is there have been work that has been done, but it needs to be completed. The work that has been done, I want to congratulate Litmus and Nimble for what they have done. And I want to say that I think, and I could be wrong, in a way or another, maybe the regulator at that time were focused on histology, and that the Litmus and Nimble finding the right NITs to replace histology was the goal. And they started outstanding protocols and procedures. For instance, the reproducibility of the test, the revalidating the test, all these things are needed to get where we at. So along the road, then the FDA started talking about, and let me go to the direct target. If you want to enroll a patient in a phase 3 study with NIT and monitor treatment response, this should be only related to outcome. And I don't know of a project that is quite designed for all that. There's also Goldmine by Rohit at UCSD that looks at MRE, but he will bring in all the NITs and putting in a big melting pot to make it a fair game, if you want to say, for everyone to see which NIT will be chosen for phase 3 studies and eventually replace the outcome, the histological outcome, with non-invasive testing. Of course, that will translate eventually into clinical practice and clinical guidance. But to close this and go to the effective talk about today, as Stephen mentioned, there are plenty of data that showed correlation with outcomes that we need a little bit more. There are data that showed the screening failure rates, placebo. So we have designed this study as retrospective and prospective. And we've been honored, both Jörn and Naeem have accepted to lead the retrospective steering committee, especially with also ideas that they were voicing around the same time we did. And there's some work that have been done by them. So we're extremely excited, and we're standing behind them to follow their lead and thoughts.
And I think you said something really important in reference to the work that's being done already in parallel, and that is highlighting Litmus, Nimble, and Goldmine. We don't want to take away anything from that work. It is incredibly important work. The data that's coming out from Litmus already in the form of very important meta-analyses, patient data meta-analyses, the work that was presented by Arun and others at the most recent Congress from Nimble is really important. Of course, the work that Rohit and UCSD and others are doing, including many of us as part of all three of those organizations, is critical. This is, to me, closing the loop. It's tying everything together. You very seldom score a touchdown in one pass. Generally speaking, you march down the field in iterative steps, gaining ground. You get into the red zone, and then you punch it in for a touchdown. This is the analogy. We're all on the same football team. We're bringing a little something different to the table, but we're all focused on one goal, and that is getting the ball across the finish line and making it easier for our patients, making it easier for clinicians to both diagnose and treat. And listen, we're on the cusp of potentially our first drug approved for the treatment of NASH. If you think about that, that's monumental. I've been at this thing for 20 years. I remember back in the early days looking at orlistat to treat this disease, simvastatin, vitamin E and vitamin C, all this work that was really after the same goal, making it easier for our patients, finding a treatment for our patients. And that's the goal of NAIL-NIT. It's not to be done in a vacuum. It's to be done with community involvement, with lots of involvement from every stakeholder that's interested in this field. And to have Jörn and Naim leading that, I think, is going to be critical.
So what exactly, Jörn, Naim, is the work that you're doing and the leadership role going to look like? And what are some of the things that you'll be going after?
As part of the clinical trials that we have been doing for the past few years, there's a lot of data that was generated that can answer many relevant questions. The problem is we operate sometimes in silos, and pharma companies are overprotective over the data for good reasons. But now we are seeing this potential to actually combine data from different pharma companies to answer the relevant questions to all of us. So one thing is, as Mazen said, all the NITs that are being done in clinical trials, can we combine data from 5, 6, 10 clinical trials and look at them and see how they perform in comparison to liver biopsy, but also in comparison to each other and some of the scores that we've developed? Can we also look at the placebo response rate in different trials and try to get a deeper understanding at what's causing this variability, which can really sometimes kill your trial if you end up with a high placebo response rate? Can we look at NASH cirrhosis trials and see what are the reasons for screen failures in these specific trials? Stephen has done a lot of work looking at reasons for screen failures in the non-cirrhotic NASH trials, but there is this unmet need to look specifically at NASH cirrhosis. So we have now this database and the willingness from different pharmaceutical companies to combine their data and try to answer some of these questions. We want to do this retrospectively first, but then of course, we want to do it prospectively and get to a point where we actually can design trials without the need for liver biopsy.
Right on, Naim, and he said it perfectly. There's so much data in the field, and it's not being analyzed with the same rigorousness from the same perspective or the same angle. That's where the power of NAIL-NIT, in particular starting out with the retrospective part here, comes in. Pulling together the baseline biopsies of these three phase 3 trials we're getting the data from, and then additional phase 2 data trials, and looking at differences, comparing this with placebo response rates. I think this will be crucially informing us moving forward.
Roger, can I jump in? So can I ask each one of you, what would be your very first project, like each one of you is the one that's passionate about, and what's your timeline for that? And I'm not asking you for a deadline for a paper or anything like that, I promise, but I'm just asking you about your passionate first project.
I'll go first, I guess. My first thing I want to do is to look at what thresholds are the ones that correspond to histologic response with different NITs. I have a special interest in corrected T1 as part of liver multi-scan and trying to validate what decrease in CT1 corresponds to improvement in the NAFLD activity score and NASH resolution. I'm also interested in looking at the MAST score and validating this in a larger database, and then looking at changes in the MAST score over time. This is Mazen's baby, but it's also dear to my heart, so I want to make sure that we validate this and look at the changes over time. Also looking at some of the scores that we generate with FibroScan. Examples are Agile 3 plus, Agile 4, and looking at longitudinal changes over time and seeing if we can use them as a way to also monitor response.
Very ambitious, Naim. Great projects.
I think you did a great job answering your question, Mazen. For somebody that's got 2 baby girls, MAST is too bold to be a girl. It's a boy. So now you have a baby boy named MAST that Naim is going to help bring into adulthood.
Yeah, sounds like a mustache.
Mazen, before Jörn answers your question, I want you to know that between some of the questions that I hear Jörn ask from time to time, the questions you're asking, I can go on vacation now.
I'm not taking over it, Roger. I promise. I'm gonna give it back to you. But I'm starting the projects on this broadcast.
We'll keep you busy, man.
Jörn, how about your babies?
Yeah, I think I do like to understand some of the differences in effect size we're seeing across trials. I'm really excited about pulling this these data sets together and looking at more than one. Now, there's some hurdles to that because slightly changed inclusion-exclusion, maybe different pathologists reading slides and so on. But I think it's something where the placebo response is planned out nicely. So I think this is one of my first aspects. And it's something I think I mentioned to Stephen sitting next to him in a litmus meeting in Spain somewhere a long time ago. We were discussing how's the placebo, how come that is different in these trials.
I think that was in Barcelona. And I think that's where I'll see you again in 2 weeks or something.
Yes, right. And see the corresponding effect size of the NITs next to that. That's really something I'm interested in.
Roger, if I may say something, hopefully no one will find this offensive, but I was reading this book called Skin in the Game by Nassim Taleb. I don't know if you're familiar with him.
Yes.
He wrote also Black Swan. He talks about these situations he calls intellectual yet idiot, okay, IYI. And the idea is that sometimes we get too intellectual that the end result is really just counterproductive. And I was thinking about it in the context of NASH. And I'll give you some examples that I really hope that NAIL-NIT will get us out of. Number one is in clinical trials, as Mazen said earlier, we have patients that show up with liver biopsy with F3, and they have steatosis 1, inflammation 1, and no ballooning, or sometimes even more inflammation. So the NAS could be 3, even 4, but there's 0 ballooning. And then we cannot enroll these patients in trials because they don't have technically NASH because they don't have ballooning.
And that patient is F3 probably too.
3, exactly. So if you think about it, Roger, the whole reason why we have NASH is because NASH is considered the aggressive progressive form of the disease. But if someone has F3, what else do you need to call it an aggressive disease, right? So now we have this situation where we cannot enroll these patients in trials. If insurance restrict to, trials and meeting the criteria for the phase 3, how can you include these patients? Do we need to do now investigator-initiated studies and include these patients in a separate trial and then not allow ballooning? It just doesn't make any sense. So I think this is one situation that NITs will help us move past and just never have to worry about ballooning on a biopsy. The second scenario is the whole NASH resolution and the requirement of ballooning 0. It just doesn't make any sense to take someone with, let's say, S2, ballooning 1, inflammation 1, and if they go to S1, ballooning 0, and inflammation 1, now we call it NASH resolution, and this is a great outcome, as opposed to someone starting with NAS of 8, let's say S3, ballooning 2, inflammation 3. If they go to S1, ballooning 1, and 0 inflammation, we cannot call it NASH resolution. I think patient 2 improved much more than patient 1, and patient 1 could have been just, pathologist variation and saying there's ballooning or not. So this is another scenario that we made it so complicated, it just doesn't make any sense. And we sometimes talk too much about evidence base and we need the data and this and that, but I don't underestimate the power of just thinking about the scenarios and saying, okay, whatever we have is just not helping us answer these scenarios and we need to use a more logical and rational approach. I think the data that will be generated in NAIL-NIT will help us get out of these IYI situations.
Naeem, that's great. And I want to thank you for teaching me one important thought about all this. And then I've got a question for you guys based on kind of my background. The thought is Naeem pointing out that we've made these rules based on no data or no realistic data, but that you need tons of data to turn them over, which would strike me as an extraordinarily good example of IYI thinking. I mean, there was an episode where you were talking about things that we've done that were proved and accepted that were never proven. But now you have to disprove them to get away from them. And the data never existed to do that in the first place. At one point you said we had better data on a lot of NITs right now than we had on biopsy when biopsy was accepted the way it was as a standard, if I recall your statement correctly. I think it's important for us to keep in mind how a lot of this stuff came about. And we talked about this a couple of weeks ago when we had the balloon hepatocyte episode. In the context of semi-quantitative and quantitative, it feels to me from the outside in happened as the ways back. People looked at things that made sense when treating individual patients, turned them into rules for larger patient populations, and statistically the rules didn't make any sense, and logically they didn't make sense across a cohort of people, even if they made sense for individuals. And I think the examples you gave are a couple of those. When we go from semi-quantitative to quantitative by making greater use of AI, we're eliminating one source of error, but it still isn't clear that we're looking at the right targets, because very smart people talking to each other can talk themselves into lots of things that don't make sense. What kinds of effort will you be able to make retrospectively to question whether these are actually the right targets in the first place? For example, using yours, Naeem, whether we just shouldn't be looking at ballooning or we should be thinking about ballooning in a very different way. How can these kinds of analyses get at that level of question?
Before they also comment, I just want to follow up on your comment, Roger and Naeem. I want to talk about also tomorrow. I mean, we talked about it a little bit, but we're complaining about the biopsy and ballooning and all this. But we all want this field to be successful. And let's say there are now 4 or 5, actually we already have 4 or 5. Let's say we have 10 MOAs that are now in phase 3, which is great because we're going to combine them in the future and control the disease. But now you have 10 MOA, the smallest trial you can do is 800 probably to 1,000 and 2,000 patients. And you're biopsying all these patients. People will not going to accept to go to biopsy study. The field will not move unless if we look at different way, which is what we're trying to do. 2, the cardiac outcomes, we kind of like shoved it under the rug for now. And we try to do it here and there, but also we need to look at that in the future phase 3 trial. Now we're too fixated on the biopsy, but as the drugs come, there will be a real need, like the type 2 diabetes field. They do trials in the thousands and thousands and look at cardiac outcome and kidney outcomes, and those are all important in our field. So that's part of the, I guess, what you said, Stephen, the touchdown approach with the football.
Some of the comments you made, Roger, are really simplistic in tone, but they're very complicated to unpack and unwind. So dating back to the advent of the FDA getting into the business of finding a drug to treat NASH, we had to come up with something. We came up with some histopathologic criteria that had modest outcome data linked with it, particularly NASH resolution. And fast forward now to where we are, we realize that those criteria are challenging to hit consistently for lots of reasons. Heterogeneity, inter- and intra-observer variability, the size of the biopsy, the staining that goes with it, the processing that goes with it, the shipping it all over the place. Some of those we've fixed, some we haven't. And now we're left with trying to come up with a better test, a non-invasive test, that we can't go back to where we were 15 years ago and just pull a marker out of the air and say, this is what we'd like to study. The authorities have said, well, it needs to link to an outcome, despite where we were when we came up with the histopathologic criteria. To me, it's a bit like this phenomenon of morphic resonance. Have you guys heard of that? Morphic resonance. It was written by a guy named Rupert Sheldrake. He's an English author, Louise, just to let you know. I think he's from Cambridge. Yeah, he's a Cambridge-trained guy. So it pains me because, there's Oxford and there's Cambridge, right? But morphic resonance is the idea of mysterious telepathy type interconnections between organisms and collective memories within species. And he gives examples of this. How do dogs know when their owners are coming home? They're in the window barking and you hadn't even turned the corner yet. Or phantom limbs. So somebody still feels their limb is attached even though it's been gone. And it's a little bit like that with where we are in the field. Naeem, you highlighted, we know this patient has bad disease. They're F3, they've got architectural changes that are consistent with a NASH phenotype, but yet we just can't bring ourselves to call it NASH by our current definition because we can't find the rogue balloon hepatocyte. But we know it's there. I think part of this is just kind of resetting, taking a step back, getting back to Louise's comment earlier, keep it simple, stupid, the KISS principle. We know these people have disease. When we did a post hoc analysis on the Intercept's Regenerate trial, I'll never forget this, when we were putting together the data that Zobairionasi presented on the phase 3 data, we said it didn't hit NASH resolution defined by ballooning of 0, inflammation of 0 to 1, but if you look at a Gestalt definition of NASH, it did hit stat sig. So here we have pathologists saying, if you just let me look at it, just like Naeem just did, if you just let me look at it, this is NASH. But if you apply strict criteria, the ones that we came up with, it wasn't NASH. That is what we need to get beyond. And my hope is that these NITs, these non-invasive testing strategies that we bring forth, whether it's the diagnosis of at-risk NASH, prognosis, or measuring therapeutic efficacy, that we're able to apply a certain amount of reason to it and understand that no test is perfect, but where we are able to define a reasonable sensitivity and specificity, that's what we take forward. And know that there's not going to be one single NIT that addresses every one of these questions. And that's the beauty of having Naeem and Jörn and the entire steering committee involved with the process, because each of us brings something unique to the table and a different perspective that could be applied toward the various NITs that are out there, whether that's MAST, MEFIB, FAST, Agile 3, Agile 4, NIST 4, you name it. Or something new, Pro C3, ELF, combination of all of that.
What happens next on this path?
Yeah, I think as Stephen said, we aim to fill the gap that is left behind or left open by the big consortia, NIMBLE, Litmus, and really trying to understand, based on the data that's there, explore the aspect, as Naeem said, where do we have the discrepancy? How can we, with the available data, close that gap? Or inform Stephen and Mazen and say, well, I think, in the prospective part, you should really have a look at this because we found this is the reason why we fail in a significant number of cases, or inform them on certain patients that do have more specific characteristics. Stephen has done that extensively, but pulling this all together, I think the power is just going to go up, and maybe even apply some sort of artificial intelligence technologies, get a partner in that looks at the data set and informs us on how to make NAIL-NIT move forward. That's from my perspective. That's how the retrospective is going to support the project.
Can I ask you a question? The data that's being collected, obviously, particularly for the retrospective, does it go as in-depth as people's presenting symptoms, what they regularly presented to the primary care physician with? Because if I look at it as a whole, NAIL-NIT, if we go backwards, should, with the numbers of patients, be able to bring us down to a questionnaire, a formation that somebody can fill in a primary care physician's and say, you are more likely to have this. These are the non-invasives that we would suggest because you scored X on looking back on that, so that we can actually nail the right NITs for the right presenting symptoms or cohort symptoms in each of the individual sort of phenotypes coming through. Do you think that's possible from combining all of you guys' work with Litmus and all of that?
That's why we love having you on the show, Louise, because you just gave me a great idea. Most of the data is generated in the context of clinical trials, but they have actually quality of life questionnaires, and it wasn't even on my radar, but now I'm going to look into it and see, because often we use the same ones, and there are a couple that were developed for other diseases. There are some specific ones for chronic liver disease and NASH specifically. So yeah, I mean, we would have to look into this and actually do it the way you said it. Look at the quality of life questions, what are the issues they're dealing with, and then look at the NITs, look at the phenotype, and see if we can actually come up with a specific set of NITs for specific presentations. I love this idea.
I love that she gave you the idea, and I love the fact that you guys are so far committed to 8 papers from what I'm counting.
But I think the whole point is most people that get to a trial are the very few of the very few of the very few, and we want the whole of the pie to be diagnosed. And that's in a primary care setting.
I have to brag on Naeem for a minute. This guy has 2 little girls. He has a clinic in Cleveland and a clinic in Phoenix. Like, for those of you who don't know geography in the U.S., they're not like right next to each other. They're like a long way away. And this guy has the ability to crank out a wonderful paper in a weekend. Like, I'm just going to sit down and just put something together. And it's like this beautiful prose that just happens. And what takes him a weekend to do might take me 6 months to do. So you might get your 8 papers, Mazen. And Jörn is very critical, and he looks at things through an eye that I don't have the ability to do. So the 2 of them, just, 8 might not be near the ceiling.
Thank you,
Stephen the whole thing is just like he wants to increase the 8 to 10 and 12. They're coming by the end of this show.
Yeah, I don't know where to take that, since I'm a painfully slow writer and I've never written an academic paper in my life.
It just struck me. Jörn, you have 2 girls too,
Yeah,
right?
that's right.
Yeah.
So all 3 of you guys, 2 girls each.
Yeah, actually,
That's a funny coincidence.
If I count in this one, I also have 2 girls. So that would make 4
Is
of us,
that
right?
true? Oh my God. Listen, I love my girls. They're the world for me. And yep, I'm all behind women power. I have to tell you, my 40 years old went to the market, and I'm sorry to hijack the talk, here in Spain. And what's that thing, like the flamingo high heels? She goes and grabbed this flamingo high heels for kids that is pink with black dots on it. And she's trying to dance flamenco, 4 years old. And she's wearing those 24/7, including going to bed.
And I bet you've got some great video.
Indeed.
That's amazing. Remember those moments.
Yeah.
The other saying I'll say is that days drag by and years fly by. Just remember that when they're 18 and leaving home.
Or when you spend the weekend with your roughly twice that age and her 2-year-old son who's named after your father.
Yeah, no doubt. No doubt. Good memories.
That was last weekend. It just keeps moving. So let's crystal ball for a minute because it's fun. We're here in a year, and then we're here in 2 years. What do you think will have come out of this work? What do you hope will have come out of this work? And how do you believe it will shape the prospective trial and the other things that people are doing outside of NAIL-NIT?
Well, let me take the global view and then I'll turn it over to the smart guys. What I want to see come out of this is something that is crystallized, a better idea of the right roadmap for how to evaluate patients with fatty liver disease. Because right now, there are any number of different pathways that you can look at, guidance documents that are out there, and some are similar, some are dramatically different. Some start with a blood-based biomarker, some start with an imaging biomarker, and if all we do is validate one of those current paradigms that we can simplify and spread throughout the world on this is the best way to go about diagnosing this particular patient cohort, that would be my dream and my goal. It might be that we come up with something totally different and unique, but my guess is we take what's out there and we look at the data we have and we try to validate one or more of those pathways, ideally one. And I think that would be a huge win. We don't have to reinvent the wheel. We just need to make it run smoother.
I really hope the retrospective part will be the engine to publish 10 to 12 papers every year. This is what we're committing to, me and Jörn, right? Because this is going to be unique, honestly, I think, in any disease state to have, several pharma companies, stakeholders agree to share data and answer all the relevant questions. But the big picture, I guess, is to have the prospective study funded and to start enrolling patients. We have big plans for this year. We are building a phase 1 unit that will have 24 beds here in Phoenix. We can have patients stay with us for a week, 10 days, do PK studies. But part of this phase 1, we're going to have our own MRI machine, all, state-of-the-art imaging. And I really hope that we will start enrolling patients for the prospective NAIL-NIT in this new unit. Stay tuned, but I think December 2022 will be up and running to do hepatic decompensation studies, PK studies, anything phase 1, and all imaging testing.
Wow. Jörn, Mazen, Louise. Louise, you and I are going to be about equally qualified to comment on this, but we'll have a go at it. But Jörn, why don't you go ahead, or Mazen?
I was thinking about, I'm now fixated on the number of the beds that you guys are going to have. 24. We're starting 10,000 square foot in Houston, Texas very soon. And we're going to have phase 1 unit, but I'm not sure about 24 bed. We're starting.
You will have an MRI right next door too. So
that's true. We're starting with 4 to 5 bed, but that's my goal. Now, my dream is to extend to 24 bed. Not that's not competition. Just
No, Mazen. You're never satisfied with equality.
No.
You need 34 beds, 40 beds.
No, I'm not that competitive. No, but truly, I'm excited about this. Next year, hopefully, we'll have good news about registry study, phase 3. Fingers crossed. Who knows what the envelope is going to open as. But at the same time, I really want to look at that 100-mile vision and predict these, hopefully, 10 to 15 MOAs in phase 3 studies while we're doing multiple investigator-initiated or phase 2 studies that is a combo now. So that's for the future. And really, to get there, we cannot just keep jabbing and stabbing people on the right side of their body with a liver biopsy. So they tell you, in residency and fellowship, what do you want to be in the next 5 years or 10 years? And I think within the next 5 years, we really have to move beyond that. I think by year 5, these guys and the rest of the steering committee, there are many exciting people and KOLs will prepare the field for those thousands of people in phase 3 without a liver biopsy and multiple phase 2 combination studies.
And just to follow up and think one step ahead, these are incredible research capacities all Mazen, Naeem, and Stephen are having. But if we accelerate this and move beyond the biopsy and have NAIL-NITs produce successful outcomes, we'll have many more study centers, even less experienced hepatologists being able to enroll their patients because they're everywhere, these patients. And I think we're going to get more diversity here in terms of participating centers.
Yeah, given how many patients are going to have to get enrolled, that makes tremendous sense. The more places you have that can enroll patients, that's huge, right? We're producing study demand faster than producing patients right now. That certainly makes sense.
As this data is generated, it's about the excitement of how to put it into real care beyond the clinical trial setting. How do we get it back into the threshold of the primary care physician, the district general hospital who's not got a liver specialist? How do we generate something that's a tool that's able to be used more locally? Because as we say, these patients are everywhere. I've recently done a catch-up FibroScan list, and I could have enrolled 5 studies in specialist centres given the limited number of slots that you get just on one session, but most of them would have probably failed biopsy. So it's really about the practical implications. I think that goes back to what Naeem was saying about we can be too intelligent for what we want to actually put it into the practical setting. So I think NAIL-NIT offers an amalgamation of that to be able to put it in practically, and that's the key for me, for more people at ground zero to be able to be diagnosed, to get the appropriate care and the appropriate NIT that they need to get them to the fastest place, quickest, because that's when we can treat them and that's when we can turn it around most. So I think it's really exciting.
That's a great point, Louise. The timeline for this, if you just want to just kind of break it down into brass tacks, Roger, we're looking at potentially, now, Intercept could resubmit any time or later this year, and maybe obeticholic acid is approved 6 months from the time of their submission. The next one up is resmetirom, and they read out in Q3 of this year. So basically Q3 of 2023, you would think that there might be a possibility of the first approval if Intercept doesn't get it before that. So there's your timeline. We need to have a very clear, easy to use, non-invasive testing strategy for at least the context of use of diagnosing the patient that would qualify for treatment in a very simple, easy to use, non-invasive format by Q3 of 2023, if not sooner.
Stephen, first of all, that sounds like a non-trivial lift, even with 8 to 10 papers coming from these guys, or 12 papers, or whatever Mazen's going to talk them into, and you by the time you're done. I want to go, the comment I made before that you said was simplistic but complex. I totally agree with you on both those things. The technology that wins isn't always the best technology, it's the technology that gets the best foothold. So what you're challenged with here to some degree is how to find a technology that can get a good enough foothold given what you're dealing with, even if it's not optimal. Another thing I think I've heard you say on a couple of occasions, and I say from time to time, is by not letting the right be the enemy of the good. It feels to me like this is a goal of getting to good.
Yeah, but what do you mean by technology, Roger?
Well, the classic example, Mazen, that they talk about in business is a long time ago with VHS, audio video cassettes, right? Sony had a much better technology called Beta, but the distribution was in favor of a clearly inferior technology called VHS. And within 3 years, VHS had swamped the market and Beta had gone away. And if you designed for the best technology for what was known at that point in time, you would have gotten to Beta. But it was about momentum and distribution and prior investment.
So let's take an example of a grant. A lot of grants, they get submitted with prelim data. A lot of times the work is already done. We're not done here. But here we have so many data that is pointing that we will get there for sure. Transitional elastography has correlation with outcome. MRE has correlation with outcome. ELF has correlation with outcomes. The needle is moving with NITs and histology. It's all there. We have just to put it together and to do that phase 3 diagnostic trial to show what the FDA wants and call it the day. So the technology is there, the data is there, it's just closing the loop and wrapping things up.
Now, that's not the 12-month process, right? That's the...
Yeah, I mean, for the people who are listening, my prediction, we will get to hopefully replacing it in 5 years completely. Now, if a magic happened before that, and data, again, I'll use the analogy of the football team. If NAIL-NIT does some and others bring more, and all of a sudden, 2024 or 2025, the FDA is saying, well, let's move on, that's even better. But I expect 5 years turnaround time to have phase 3 completely, 5 to 6 completely, depending on using NIT. What do you think, Stephen, Naeem, and Jörn? Our plan is 5 to 6 years. So is that too ambitious or the other way around?
No, I don't think it's too ambitious. I think, quite frankly, we have to have this within 5 years, if not sooner. If you look at clinical trials, we can enroll a non-invasive test 10 times faster than we can enroll a paired liver biopsy study. In essence, it's the same patient population often, but because of the complexities of liver biopsy, the high screen fail rate, and the fact that patients often don't want to go through it, it's led to inevitable delays in drug development. Without drug development, we can't get after disease awareness. Without disease awareness, we're not going to get after the patients that have the disease. disease. So it's a vicious cycle. We have to break it, and we break it by finding a non-invasive test. That then frees up clinical trials. It allows drugs to be developed. It allows funds to come in to build disease awareness campaigns. And then we are able to get after combination therapies, just like hepatitis C. And one day we're talking about eradicating the disease rather than finding our first treatment for the disease. We don't have that within 5 years. I think that the momentum we've gained will be lost. The interest in the field will have moved on, and we're going to be in a bad spot.
I think the sad thing is we're using a method in liver biopsy that's obstructive to patient survival. Now, that should never be allowed, that using biopsy obstructs patient survival and timelines. And I don't know any other disease that would be placing that as a barrier.
Yeah, and based on the homework, Mazen, you gave to us, I mean, there's a lot of data in regulatory trials already being collected, and we're going to be busy with that. But I think I echo Stephen. We can be more ambitious because we've already done so much in these regulatory trials. And just to have another thought here, it's not going to be a different disease once we use NITs to assess it compared to histology. But I think we'll reach different patients, the ones that pulled out before because they didn't want to go the biopsy. So I think that's so crucial, really, to get to the next level here. And again, many things have already been done. Over Limbal, Litmus, and NAIL-NIT, I think we're able to move the field more rapidly.
So in a year, it'll be third anniversary podcast. We'll get back together. We'll do noisemakers. Technology will be better, so we'll be able to do more interesting things on it. We'll have a new bunch of Harrisonisms, even if you're only on for half the podcast, Stephen. I know how fast you create them. I'm sure there'll be more of them. And hopefully you guys will push this ball way further up the field. In the meantime, happy anniversary podcast. Jörn, welcome aboard full-time. Stephen, glad you're going to be around as much as you are. And Naeem and Mazen, thanks for joining us. And Louise, you get some special dispensation in Barcelona for having done this at 3:30 in the morning. We will all be together. Virtually all be together.
And hopefully the sun shines in Sydney for the rest of my time.
That would be a good thing. I'll be back with business in a couple of minutes. Thank you.
Always a pleasure being here.
Thanks, Roger. Bye, guys.
Thank you.
Bye-bye now.
And now for the Season 3, Episode 19 Business Report.
To our Ukrainian friends and colleagues, Slava Ukraini, Heroyam Slava, glory to Ukraine, glory to the heroes. As the pictures and stories of unspeakable war crimes become more frequent and more graphic daily, we become increasingly horrified by what we see and read. And to any of our listeners living under tightly controlled propaganda and propagandistic media, your media lies. These are not crisis actors. This is not something Ukrainians in the West are doing to destroy the Russian way of life. The pictures and the horrors are real. And with that, let me shift to the business of the podcast. We announced several important changes at the beginning of this episode. Let's unpack them a little bit.
Welcome to SurfingNASH.com, the corporation.
As I mentioned on the podcast, we have officially formed SurfingNASH.com as a corporation to shepherd this podcast and all our other medical communication activities forward. For you, the listener, it means we will create new media and communications to educate and inform about fatty liver diseases, and with that, put an ever-increasingly large dent in fatty liver disease. For those of you who wish to advertise or sponsor or collaborate with us, it means we will have an array of ways to work together in pursuing your communication goals. Let me know so we can talk.
Congratulations to Stephen and Jörn.
For those who've been listening recently, it's not a surprise that Stephen's schedule had already encroached on his ability to be with us every week, nor that Jörn has become an increasingly major presence on the podcast, hence the nickname Fifth Beatle. In a sense, all that we're doing today is recognizing what has become our reality. Now, with that said, the only other comment I want to make about this shift is that we recognize that 60-ish% of the audience for this podcast lives in North America. We intend to have a fourth U.S.-based key opinion leader on every episode, Stephen or Mazen or Naeem or Manal or Alina, Alan, or any of the other folks who've been with us, and we're blessed with a wealth of excellent choices. Keep listening as this develops.
Media partnership information coming soon.
We are developing sponsorship and content opportunities for all three of our second quarter media events, the Innovations in NAFLD Care 22 Conference, the 5th Global NASH Conference, and EASL's International Liver Congress. I'll announce more about the Global NASH and EASL programs in weeks to come. If you want information privately, send me a note at questions@surfingnash.com or roger.green@hepdynamics.com until we set up the Surfing NASH website fully, and I will get back to you and we can talk, or I can share information with you.
Let's talk about future episodes.
It's embarrassing, but we're still working on next week's program ideas after plans A and B both fell through. Trust us, though, whatever we do, it'll be excellent. It always seems to be. On April 25th, we will preview the 5th Global NASH Congress. The most recent draft of the agenda for that conference has some truly fantastic presentations. We're excited to be part of it. May 2nd will be an episode on the all-encompassing and complex nature of NASH patient advocacy today, or fatty liver patient advocacy today, with Andrew Scott from GLI and Achim Kautz, a German patient advocate with a fascinating and complex view of advocacy. People like Andrew and Achim play a big part in putting a dent in fatty liver disease, as do Donna Cryer, Jeff McIntyre, Olivia Alameda, other friends who've joined us on previous episodes. This conversation should be fascinating. Later in the month, we have a session about NITs with Quentin Anstey, a drug development session with more details on both to come, and at the end of the month, 5th Global NASH Congress. June has International NASH Day, EASL, a couple of other topics. We've got a lot going on.
More buzz and some new material from the vault.
Okay, on Buzz, this is now our 6th straight week of over 1,000 downloads, and our 13th of the last 16. To put that in context, before that, we only had 4 weeks over 1,000 in the first 19 months of the podcast. Last week, I reported that there was not very much interest in listening to oldies in the vault. This week, there was some. Specifically, interest in Conversations 57.1 and 62.1, and in episode 62. They're all kind of related. 57.1 was talking about how to get past biopsy to make NITs the gold standard. 62.1 was Mazen Noordien's year-end conversation on how we use digital pathology as an AI as part of how we make the transition. Included him and also Manal Abdulmalik talking about treating patients with currently available medications, and Wayne Eskridge reviewing the SUN study. More recently, episodes 14 on ballooned hepatocyte, 15 on the CLDF wrap-up, and 16 on data modeling continue to grow nicely. And the 2 recent extrasodes, 14.5 from HistoIndex on digital pathology and 16.5 from Resounding on broader use of MRE, both showed upticks last week. And with that, we're done. I have a longer than usual list of thanks, though. Thanks to Louise for waking up at 3 in the morning in Australia to do this episode, and to Mazen and Naeem for being their usual fantastic selves. Congrats one more time to Stephen and Jörn. And of course, and as always, I want to thank our team, Magic Mike, Eric, Steve, Murph the Magnificent, for your usual excellent work. Someone on the podcast, I think it was Jörn, commented on how much better the podcast this team produces every week sounds in the recording sessions that create them. True enough. I live in marvel of you guys. Finally, thanks to you, the listeners, whose committed listening and loyalty make SurfingNASH.com, the corporation, possible, and increase the size of the dent we can put in fatty liver disease weekly through listening, sharing with what you hear with your colleagues, your friends, and your family. And with that, I'm off. Stay safe, surf on. I look forward to seeing you next week again on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.